back, we had started biotic resistance in GMO and non-GMO crops. And we will have Professor Tapan Adhya to chair the session, and Professor K.K. Jena as a co-chair. And we have two speakers in this session. So here, Professor Adhya. Yeah.
Good afternoon. Welcome to session 3 continuation of the presentations. So in this talk now, it will be given by Dr. RajKumar Joshi. Dr. RajKumar Joshi is the associate professor from the University of Rama Devi University, Bhubaneswar. Dr. Raj is working on working in this university as a head of biotechnology in Rama Devi University, and he is leading functional biotechnology area in the Department of Biotechnology. So today, Dr. Joshi will be presenting on the topic CRISPR-Cas9 in crop improvement, and he will give a case study towards development of anthracnose resistance in pepper. So I welcome Dr. Raj Joshi. Yes. So we'll be giving you 20 minutes for presentation.
Good afternoon, ladies and gentlemen. On the outside, I must thank Professor Jena and Dr. T.K. Adhya. They are, in fact, some of my earlier inspiration why I started working on agriculture. So thanks to them, and I feel glad to speak in front of them today. So we have been working in CRISPR-Cas system for last 5 years now, thanks to my postdoc tenure in University of Alberta and subsequent collaboration with the Chinese Academy of Agriculture Sciences. And being a vegetable biotechnologist, I thought that why not use it on the area of molecular plant microbe interaction, which is a very important part required for improving vegetable crops. And pepper anthracnose interaction is one of the most important aspect of biotic stresses in pepper plant, owing to the fact that it accounts for 30% of yield losses, subsequently amounting to about nearly $500 million yield losses annually. So, okay, so this is the basic pathosystem. How does it work? I just give a brief idea. It is basically caused by a Coleotrichum species complex. It's not just a single species which causes the disease. So sometimes developing resistance is a tricky job here. So nevertheless, there have been certain attempts to carry out management of the disease by making use of fungicides and biocontrol agents. Also, there have been certain molecular markers developed through host resistance breeding, like QTLs, and a couple of qualitative genes are also available. But most of these things, what I'm telling, are not 100% effective, owing to the fact that severe environmental toxicity plays a very important role in this infection. There is variability in the resistance response of 3 different pathogens which contribute into the interaction. And there are also no qualitative number of natural resistance sources which could be exploited for this purpose. So can we think beyond our gene is the question in our hand. So to pursue on this part, we have done a couple of comparative analysis to see what is the magnitude of change in the expression patterns of different kind of genes during a compatible and incompatible interaction between pepper and anthracnose. And as you can see in this picture, a wide range of genetic factors play a role in ensuring disease resistance in pepper, such as the genes associated with JA and ET signaling, a wide range of transcription factors, also secondary metabolites like glucosinolates. A very interesting thing, what you can mark it here in this slide is that there are two genes, ERF28 and NAC72, which are actually downregulated during a pathogenic infection situation, meaning that these are some of the genes which might be acting as a susceptibility factor, possibly allowing the pathogen to grow in the implanter situation, causing the incidence of the disease. So they were very interesting to us. So we thought of why not further work on them. We did a couple of further experimentation in this area. Sorry, I think I'm unable to change it. Yeah. So just to prove that this is indeed a susceptibility factor gene, we did a comparative analysis of the overexpression lines as well as the silenced line. As you can see in this picture, a couple of overexpression lines are tremendously expressed, and the virus-induced gene silencing lines did not, so almost nil, negligible infection rates. And so as this correlation and the relative fungal growth is also significantly different, clearly suggesting that indeed ERF28, which is the gene I'm talking here, is a negative regulator of anthracnose resistance in chili pepper. So this was found to be a very interesting gene in our hand, which could be possibly exploited to ensure a CRISPR-Cas9 system could be developed to facilitate an anthracnose resistance pepper line development. So continuing with that, this is just a brief idea. You all know that CRISPR-Cas9 has been there in the market for the last 5 to 7 years. And I think almost every sphere of biotechnological research comes across CRISPR-Cas9 now. We talk about human, plant, microbes. Anywhere this technology is rigorously being used or exploited to bring about improvement. And these are the 3 sequence-specific nucleases which are available, of which CRISPR-Cas9 is the most wisely used one, considering that it does not use complex chemistry, can be easily joined, made up, and without much difficulty. And this is just a representation, how CRISPR-Cas publications are influencing us during the last 5 years. You can see more than 10% of the most cited publications make use of the CRISPR-Cas technology. So that itself says how influencing the technology is. And it is also evident from the fact that the 2 inventors of the technology received the Nobel Prize within a very short period of time, considering the significance of the work. So taking that into account, we thought of designing a CRISPR-Cas system to ensure anthracnose resistance in chili pepper. And here we tried developing a very unique technology, that is the single transcript unit based system. Generally, most of the CRISPR-Cas technology that is available in the market make use of a dual promoter system, because we have a Cas9 protein from the bacteria and a RNA sequence taken out from the eukaryotic organism. However, this single transcript unit system make use of a single polymerase II RNA polymerase promoter, which regulate the expression of both Cas9 as well as the sgRNA. And we do have a hammerhead riboenzyme sequence, which facilitate the breakage of the transcript, separating the Cas9 and the sgRNA subsequently after transcription, so that it generates the complex which can go and carry out the cleavage action. So based on this background, we designed a couple of constructs. ERF28 is a 2800 nucleotide gene having a single exon, and we designed a couple of guide RNA sequences from the exonic region, meaning that we can possibly facilitate prevent protein expression. So these are the 2 constructs that we designed. The general method of transformation, I don't want to explain more on this. We used the hypocotyle of the plant, and an agrobacter mediated gene transfer was used. This is interesting. As you can see in this picture, we ended up with about 30 to 40 transgenic lines, out of which about 70 to 80% of them were mutated at the desired targeted gene. And all the 4 possible mutant genotypes were realized. And among them, the biallelic mutants were the most highly represented one. And among them, the deletion mutants were the maximum one, which were to the tune of 65 to 60%, 60 to 65%, sorry. And we also designed a construct where we inserted both this SGRN together, making a single S1, S2 complex, and found that the percentage of mutant efficiency increases. So we concluded that if we design construct having multiple targets associated, the mutation efficiency can be significantly induced, and we can get the mutant quite easily. Moving ahead, the most important thing is that once we get the mutant, it needs to be inherited following the basic Mendelian principle, to be very frank. And we also tried to do that. And initially in the first generation, we ended up with only a few number of homozygous lines. But then subsequently, we took those homozygous lines and tried to do this aggregation. And we found that all those mutants that were generated in the T0 generation subsequently moved into T1 as well as to T2. And the transmission rate was 100%, absolutely. So this clearly suggests that CRISPR-Cas9 mediated mutant generation also follows the basic Mendelian inheritance pattern, and the mutation can be transmitted subsequently into following generations. A very important thing which we need also to take care of when we use CRISPR-Cas9 system is that we have to make it transgene free. Always regulatory issues comes into picture when a plant is transgenic, and no doubt the tDNA what we use is transgenic. So we also did analysis of the Cas9 protein. Just wanted to see which are the plant or the mutant line which did not have a Cas9 protein, so that we could take them further, considering them as the Cas9 free plants, and carry out the expression. So we used a pair of markers which could be possibly used for finding out which are the plants having the Cas9 proteins. As you can see, there were some few number of bands not available, not visible in this picture, suggesting that those are those plants which were free from the Cas9 protein. So we just targeted those group of lines and carried forward with our experiment. So overall, we selected about 5 homozygous mutant lines which were Cas9 free, meaning they were not transgenic, they were only the mutated one, and confirmed them, so carried out certain validation. On the left-hand side, we also perform a T7 endonuclease assay, which is a detection system for seeing whether Cas9 mediated cleavage has been realized or not. And those lines which are mutated at the target sign gives you multiple fragments, as you can see here, and the wild type does not break because no heteroduplexes are created. And we also did a time score expression analysis. As you can see, while the wild type had a tremendous increase in the transcript accumulation level of the ERF28 gene in the normal plants, while in the 5 mutant lines what we selected, I mean, we have a complete negligible expression pattern. The same was corroborated with the protein expression. I mean, there was no expression of the protein in the mutant line, meaning that the gene is transcriptionally silent and there was no expression of the protein realized. Whereas in the wild type, we indeed got a distinctive ERF29 protein fragment. Then the phenotypic assay, as you can see, if you compare the wild type with that of the 5 mutant lines in here, there is a very negligible pathogenic growth in the ERF28 mutant lines, while in the wild type, there is a significant pathogenic load. And that can also be corroborated with the fact that the lesion growth, the sporulation, and the relative fungal growth was also significantly diminished due to the mutation of the ERF28 transcription factor in the mutant plants. Another aspect what we wanted to study here is to see whether the ERF28 modification is indeed facilitating a defense response. So we just saw that anthracnose growth has been diminished, but we also wanted to see whether this ERF28 modification facilitated any kind of change in the defense mechanism. And as you can see here, we selected a couple of defense responsive genes, and you can see in all the mutant line, there is a significant upregulation of some of the most prominent defense responsive genes, taken into account the lipoxygenase, the PR proteins, the plant defensin proteins, and many of them. So suggesting the fact that indeed the mutation of the ERF28 also facilitated enhancing the defense responsive mechanism by some way or the other. We haven't yet found out what are the different mechanisms directly or indirectly modified by this process, but this is one of the link that we could prove. Off-target modification is another factor we need to look into when we work on CRISPR-Cas9 system, because sequence similarity may also result in cleavage of unwanted genes, or maybe some functional genes which might create other kind of problems in the mutants. And in this case, we also found about 4 off-target genes, but mismatches were less than 3. That is what is allowed. And just to prove that thing, we took all the 5 selected mutant lines and carried out amplification cum sequencing of all the 4 off-target genes, and we found that it was less than 5% of target modification, which is within the acceptable level. If you are doing an SDN1 experiment where just a knockout is being carried out, I think 3 to 5% is acceptable globally. So overall, I could conclude the targeted mutation of ERF28 indeed enhances the resistance to anthracnose in chili pepper. And this single transcript unit CRISPR-Cas system, what we have used in this method, is indeed a very efficient method. And there are more advanced version of this single transcript unit which is available with us. We have replaced the polymerase II promoter with a tRNA polymerase promoter, which is more efficient in targeting a specific gene in vegetable plants. And if you talk about the future activity, we are trying to see the modification. See, mind you, when we did this experiment, we specifically targeted the most belligerent anthracnose pathogen, that is the Coleotrichum truncata. And the disease is actually caused by a complex of 3 species. So we are currently trying to see what is the nature of expression or defense that is being realized by this mutant line against all the 3 pathogens. Then only a broad spectrum anthracnose resistance in pepper could be realized. And also we are trying to see if there is any change in the efficiency when we use a dual promoter-based CRISPR system and a single transcript unit-based CRISPR system. So as to say or prove that we can gradually replace the dual promoter CRISPR-Cas9 system, which is indeed a conversion process of designing 2 different vectors, putting them together. Instead of replacing that with a single ready-to-use vector, you just purchase the vector from the market, insert your sgRNA and just do the transformation. So before I end up, I have certain challenges and limitations to discuss. What I'm just trying to tell you here is that CRISPR-Cas vector system make use of nucleotide mutation or fragmental deletion to NHEJ or double-stranded break mechanism. While if you talk about the NHEJ, it is very well established in the plant system. We can carry out it very easily. We can select the mutants very nicely. But if we talk about the homologous recombination part, it is always a tricky thing in plant system. So we need to develop CRISPR-Cas system to make it more efficient through a homologous recombination based directed repair, because that can solve a majority of agronomic problem, because you all know that single base variations are primarily responsible for large number of allelic trait modification in plant system. So if we can streamline and strengthen the homology directed DNA repair mechanism to use it in CRISPR-Cas, it would be a real boon. And the Chinese have already started doing it in a very rapid manner, and we are also following through. So, and base editing is also recently emerging, which could also compensate the homologous recombination. If it is not possible in a plant, we can also go for a base editor and carry out the experiment as well. So this is the group. Professor Zhao is from Chinese Academy of Agriculture Science. He has been a collaborator in the vegetable research part. And Professor Nat Kev is from University of Alberta, and he's contributing to us in pathogenic interaction part. And the other guys have also significantly contributed. Vijaya is the current PhD student. Thank you so much.